Hello everyone. In the previous video, we have seen introduction for the process photosynthesis. In this video, we are going to see different raw materials that are needed for photosynthesis and how with certain experiments we can prove it. If you look at this process, it is a biochemical process that happens in plants, that too inside their cells. So in order to identify that, we should understand this equation. Only if the raw materials or the reactants are there will be occurrence of the process, and only if the process occurs, there will be formation of the products. If you see these products of photosynthesis, water will be there in almost all cells, and as far as oxygen is concerned, if oxygen is needed for respiration, mitochondria will be utilizing this oxygen. But if oxygen is in excess, it will be let into the atmosphere by the plants. So the most reliable product to detect this process is glucose. When cells need energy, they will utilize this glucose during cellular respiration. When this glucose is in excess, it will be converted into a storage form known as starch. By detecting starch, we can easily identify whether the process of photosynthesis has occurred in a particular region of the plant or not. Starch can be easily detected using iodine solution. Iodine solution can be prepared by mixing iodine crystals and potassium iodide in distilled water. If you look at these photographs, both of them represent starch grains of potato cells. These starch grains are unstained starch grains, whereas these are stained starch grains. If starch grains are stained with iodine solution, they will take up this dark blue or bluish black color. If you look at this picture, only starch grain alone is taking up the color, but if a particular organ of the plant is detected for starch, and if that organ contains starch, organ will take up this color. With that, we can easily say whether photosynthesis has occurred in that particular region of the plant or not. The experiments that you are going to see here are already done by you. We are just going to recall it quickly. Chlorophyll, light and carbon dioxide, all the three are very essential for photosynthesis to happen. In order to prove that chlorophyll and light are essential for this process, we are going to use variegated leaf experiment. And to prove carbon dioxide is essential for photosynthesis, we are going to use moles half-leaf experiment. First, let's see experiment which proves chlorophyll is essential for photosynthesis. For that, we require a variegated leaf. If you look at this leaf, there are patches of pigments. There are certain areas that are green in color. In the same leaf, there are certain areas which are non-green in color. Those that are green in color represents presence of chlorophyll, whereas those that are non-green in color shows absence of chlorophyll. Why we have to take this kind of leaf for this particular experiment is, we are going to observe whether photosynthesis occurs only in the region where chlorophyll is present or it occurs even in the region where chlorophyll is absent. How we can detect that is by using iodine solution. If starch grains are seen only in the region where chlorophyll is present, we can easily prove that chlorophyll is essential for photosynthesis. As we are going to prove the experiment only by the presence of starch grains, we should be 100% sure that there are no starch grains that are already present in the plant. So before starting the experiment, we have to de-starch the plant. That is, we have to remove the starch grains in the plant. This can be done by keeping the potted plant in dark conditions for 2 to 3 days. At least it should be kept in the dark for 24 hours. Only then we can use that plant for this particular experiment. When we keep the plant in the dark, starch grains stored in different parts of the plant gets utilized because in the dark condition they cannot do photosynthesis, so they utilize the storage form of carbohydrate. Now this plant has to be kept in sunlight so that it can start photosynthesis. When these plants are kept in sunlight for few to several hours, photosynthesis will be happening. After the incubation in sunlight, the leaves can be removed from the potted plant and can be tested for starch using iodine solution. But before putting that in the iodine solution, different pigments in the leaves have to be removed because these pigments can interfere with the iodine solution. So in order to remove these pigments, we have to keep these detached leaves in boiling alcohol, which is kept in boiling water bath. Once we keep these leaves in boiling alcohol, the pigments will enter into the alcoholic solution. Leaves will become transparent. But the leaves will also show some shrinkage because alcohol has the tendency to remove water as well. So these leaves that are colorless has to be rehydrated so that it can remain intact. Rehydration can be done by keeping these leaves in hot water for some time. Now these leaves can be stained with iodine solution to detect starch. If you observe the result, wherever the leaf was originally green, that is the region where chlorophyll was present, that is the region where there is presence of starch grains, indicating that chlorophyll is essential for photosynthesis. Now let's see the experiment which proves light is essential for photosynthesis. For this experiment also, we have to take potted plant with variegated leaves. This should be de-starched by keeping that in dark for 2 to 3 days, and then only it should be exposed to sunlight. But this time when we expose that to sunlight, we should take care that few leaves are exposed to sunlight, whereas few leaves are covered so that they are not exposed to sunlight. The reason here is we want to find the impact of light on the process photosynthesis. So after incubating that in sunlight for some time, we have to remove one of the exposed and unexposed leaf, and then we are going to see the presence of starch grains using iodine solution. But before adding iodine solution, as we have done in the previous experiment, we have to remove pigments that were present in the leaves by keeping that in hot alcohol, and then we have to rehydrate by keeping those leaves in hot water, and then only we have to stain that with iodine solution. If you observe the results, only those leaves that were exposed to sunlight alone shows presence of starch grains, indicating that light is essential for photosynthesis. Now let's see mole's half-leaf experiment, which proves carbon dioxide is essential for photosynthesis. For this, we are going to take a potted plant which contains normal green leaves. They are not variegated leaves, they are normal green leaves. Before starting the experiment, we have to de-starch this plant by keeping that in dark for 2 to 3 days so that starch grains that were already present will be removed by its various functions. Now this plant has to be exposed to sunlight so that photosynthesis is allowed to happen. While keeping that in the sunlight, one of the leaf should be inserted into a container. This can be a test tube or any glass tube. While inserting the leaf, we should take care that one half of the leaf remains outside the container, whereas the other half remains inside the container. See, if you remember the previous experiment, in order to prove importance of chlorophyll in photosynthesis, we have taken a leaf where some of the region contains chlorophyll and some of the regions do not contain chlorophyll, in order to compare the role of chlorophyll. Here what we are going to do is, in one leaf, one half is exposed to carbon dioxide and the other half is not allowed to come in contact with carbon dioxide. That is possible because of the presence of potassium hydroxide in that glass container. This potassium hydroxide has the ability to absorb carbon dioxide so that this part of the leaf cannot have access to carbon dioxide. After incubating the entire setup in sunlight for some time, this leaf has to be removed and it should be tested for starch. But before adding iodine solution, the pigments in the leaf has to be removed as done earlier, and then it should be rehydrated and then only it should be tested for starch. If you observe the result, the half of the leaf which was outside the container, which is in contact with outside carbon dioxide, shows positive result for starch, whereas that half of the leaf which was inserted into the container, which is devoid of carbon dioxide, do not show the presence of starch, indicating that carbon dioxide is essential for photosynthesis. That is all for this video. Hope you were able to recollect the experiments that you have done already. In the next video, we will see different experiments that were done by different scientists in order to study the process of photosynthesis.